Greetings everyone. Welcome to a new episode of Mr. Bioinformatics. In the last episode, we talked about the evolution of the 3D structure of proteins. But today, we will talk about the most advanced protein prediction tool, which is Alphafold. In 2018, DeepMind, a subsidiary of Alphabet, introduced Alphafold 1, an AI system designed to predict the 3D structure of proteins from their amino acid sequences. But how? AlphaFolds use deep learning algorithms, offered an AlphaFold that drastically reduce time and effort required to obtain accurate protein structure predictions. And in July 2021, we were all surprised when DeepMind released AlphaFold 2. But why? AlphaFold 2 not only make very accurate predictions, but also does so in just a few hours. Unlike traditional methods that could take months. How it does that? The process begins with the input of a protein's amino acid sequence. AlphaFold 2 then searches in a sequence database, like UniProt, to find similar protein sequences. From these similar sequences, AlphaFold 2 then generates a multiple sequence alignment, or MSA. What is MSA? MSA is a method used to align several biological sequences to find similarities. This step is crucial, but why? Because it allows AlphaFold 2 to predict a protein's 3D structure by comparing its amino acid sequence with similar ones, revealing patterns that guide how the protein will fold. Imagine scientists want to predict the 3D structure of a new protein. If the new protein has a sequence similar to hemoglobin, for example, MSA can highlight key regions involved in oxygen binding. Since hemoglobin's structure is already known, the comparison helps predict the new protein's shape and function. The generated MSA is then analyzed by Evoformer. What is Evoformer and what it does? Evoformer looks at how certain amino acids change together, which helps AlphaFold 2 understand how different parts of the protein interacts and fold. In simpler terms, Evoformer is like a pattern detector that finds which pieces of the protein work together to keep the structure stable. What does we mean by keeping the structure stable? Protein is made up of a chain of amino acids, and these amino acids can mutate or change over time due to factors like genetic differences. When mutations happen, the amino acids might change in a way that could affect the protein's shape or function. Evoformer helps AlphaFold 2 understand how these changes. These abilities made AlphaFold 2 be used for mutation analysis to know how changes in protein's sequence can affect its function and also interactions with other molecules. This has profound implications for studying diseases and designing new drugs. For example, AlphaFold 2 was used to study mutations in the human's bioinformatics protein, which is linked to Parkinsonism disease, providing valuable insights into the disease mechanism. Not only biological research, also useful in the environmental research. For example, researchers used it to identify 74 enzymes capable of breaking down PET plastic, analyzing their 3D structures and mechanisms. PET stands for polyethylene terephthalate, a type of plastic commonly used in products like bottles, clothing, and packaging. What an eco-friendly protein structure finder. Not only that, but AlphaFolds could solve a problem that had puzzled scientists for over 10 years, which is antibiotic resistance. What is antibiotic resistance? Antibiotic resistance happens when bacteria change and stop antibiotics from working. It's like the bacteria get used to the antibiotic and develop weapons to defend themselves, making infections harder to treat. According to the Centers for Disease Control, or CDC, more than 2.8 million antimicrobial resistant infections occur in the US each year, and more than 35,000 people die as a result. With few new antibiotics being developed, researchers are focused on enzymes that help bacteria block antibiotics. These enzymes are harder to study, but AlphaFold quickly created an accurate model of one key enzyme. Someone might ask, but how do scientists know if AlphaFold's predictions are accurate? That's a great question. AlphaFold was trained on data from the Protein Data Bank, or PDB, which contains over 200,000 experimentally determined macromolecules molecular structures. To check the accuracy of the protein predictions, scientists rely on confidence metrics like PLDDT, or Predicted Local Distance Difference Test, and PAE, or Predicted Aligned Error. What are they? Well, PLDDT measures how confident AlphaFold is about the predicted shape of each part of the protein. A higher score means that AlphaFold is more than certain that the structure is correct, while a lower score means that there is more uncertainty. While PAE, it evaluates how confident AlphaFold is about the relative positions of amino acids. Low PAE means AlphaFold is sure about their alignment, but high PAE signals uncertainty. So how do these metrics help scientists determine the reliability of a prediction? In 2020, AlphaFold achieved a historic milestone by winning the CASP14 competition. What is CASP? CASP, or Critical Assessment of Structure Prediction, is a key benchmark in the field where researchers submit their predicted protein structures, and the true structures are determined using experimental methods like X-ray crystallography, NMR, or cryo-EM. If you want to know what are these methods, you can watch our episode about the 3D protein structures. These real structures are kept secret until after predictions are submitted. Once the predictions are made, they are compared to the experimentally determined structures to evaluate the accuracy. On 15th of July 2021, Alphafold 2 was officially introduced. If you want to know how advanced Alphafold 2 is, its predictions during CASP14 closely match the experimental structures. It's about 88% of the time, setting a new standard in the protein's structure prediction. Someone might ask, but how scientists compare the predicted and the experimental structures? To compare them, scientists commonly use the root mean square deviation, or RMSD. This metric measures the average distance for deviation between the corresponding atoms when two similar structures are superimposed. In simpler terms, RMSD, it tells us how far apart the atoms in one structure are from the atoms in another structure. For example, if two protein structures are very similar, the RMSD will be small, meaning the atoms are closely to each other. If the structures differ more, the RMSD value will be larger, indicating a great distances between the atoms. We know that our group or side chain of amino acid are what distinguish one amino acid from another. Therefore, any change to an R group results in a different amino acid with unique properties. To provide further detail on Alphafold's 2 accuracy, approximately 93% of its side chain predictions are close to the experimentally determined structures, and 80% are exact match. In comparison, experimental methods achieved about 98% accuracy for side chains, and 94% being an exact match. Someone might ask, does Alphafold 2 have limitations? Although Alphafold 2 is an impressive tool, it has several limitations. It struggles with predicting intrinsically disordered regions, or IDRs, which are flexible or unstructured part of proteins that play a critical role in biological functions. This limits its ability to fully capture a protein's behavior. Alphafold also has difficulty modeling protein DNA or RNA interactions, which are vital for processes like gene expression, hindering its use in genomic research. While excellent at predicting protein structures, it's less accurate in modeling ligand binding. What are ligands? Ligands are small molecules, such as drugs or ions, that binds to proteins to alter their function, making this prediction important in drug discovery. Additionally, it doesn't yet predict post-translational modifications, or PTMS, such as phosphorylation and glucosylation, which are key in protein regulations. Its predictions are less reliable for membrane proteins, which have complex structures embedded in cell membranes. Despite these drawbacks, Alphafold 2 impact cannot be denied. In 2024, Demis Hassabis and Dr. John Jumper were awarded Nobel Prize in Chemistry for their work developing Alphafold. David Baker was also co-awarded for his work on computational protein design. On 8th of May 2024, Alphafold 3 was co-developed by Google's DeepMind and Isomorphic Labs, and it came to solve many problems. Alphafold 3 is not limited to single-chain proteins, as it can also predict the structures of protein complex with DNA, RNA, post-translational modifications, and selected ligands and ions. molecules. But how Alphafold 3 does all of that? Alphafold 3 utilizes the Performer, a transformer-inspired architecture that simplifies the handling of pairwise relationships compared to Alphafold's 2 Evoformer. This simplification in part enables Alphafold 3 to expand its prediction capabilities beyond single proteins, now including interactions with other molecules like DNA, RNA, and ligands. And that's why ChatGPT has its T, as it comes from Transformer also. For deeper understanding of the Transformer architecture, you can check out our episode Gemini. In the context of Alphafold 3, instead of working with weights, the Transformer looks at the amino acids and understand how these amino acids relate to each other to predict how the protein will fold into 3D shape. So instead of relying on large amount of protein data or MSAs, it processes both single amino acid data and the relationship between pairs of amino acid directly. This makes the process faster and easier. Once the Performer makes its initial prediction, a diffusion model fine-tunes it. The model starts with a rough cloud-like structure of atoms and slowly adjusts them to make the protein structure more accurate. Tell me in the comment if you want a tutorial about Alphafold. And here we've reached the end of our episode. Thanks for watching. See you next time.